Some plants, including major crops as corn, sorghum, sugarcane, millet, and amaranth, conduct an additional mechanism, C4 carbon fixation. In the leaf of a C4 plant, carbon dioxide enters a mesophyll cell, dissolves in water, and forms bicarbonate. The enzyme PEP carboxylase catalyzes the reaction of this bicarbonate with a 3-carbon acid phosphoenolpyruvate to produce a 4-carbon acid oxaloacetate, hence the name C4 carbon fixation. Atmospheric oxygen concentrations have no effect on this reaction. Subsequently, oxaloacetate is converted into another 4-carbon acid, malate or aspartate, depending on species, which diffuses to the bundle sheath, a structure near the middle of a leaf. In the bundle sheath, malate or aspartate splits into carbon dioxide and a 3-carbon acid pyruvate or alanine. This releases carbon dioxide in the bundle sheath to concentrations of about 0.1% carbon dioxide, and a standard carbon fixation via rubisco proceeds in bundle sheath chloroplasts with negligible photorespiration. Costs versus benefits of C4 carbon fixation depends on environmental conditions. One cost is that the additional chemical reactions in this cycle expend 2 ATPs per carbon dioxide molecule pumped from the mesophyll to the bundle sheath. Moreover, C4 carbon fixation requires special anatomical features such as tightly packed bundle sheath cells to maintain large concentration differences in carbon dioxide within various parts of the plant. The benefits of C4 carbon fixation are that low atmospheric carbon dioxide concentrations or high temperatures do not inhibit photosynthetic sugar production as severely as in C3 plants. At current atmospheric carbon dioxide concentrations of 380 parts per million, the crossover point where C3 and C4 plants are equally efficient in converting light into chemical energy, quantum yield, falls between 20 and 30 degrees Celsius. Why should you care about this stuff? The physiology of photosynthesis, outlined in the preceding, determines where your food is grown today and how locations may shift with climate change. In particular, differences between the two carbon fixation pathways dictate that low atmospheric carbon dioxide concentrations and high daytime growing temperatures favor C4 plants over C3 plants. Geographical distributions of C3 and C4 plants reflect these physiological differences. For example, temperature optima of the two pathways determine the location of spring wheat and temperate corn belts in the Great Plains of North America, Europe, and Asia. The colder northern latitudes are more suitable for wheat production, and the warmer southern latitudes are more suitable for corn production. Distributions of native C3 and C4 grasses in the Great Plains are also consistent with such considerations. Global climate models predict that both atmospheric carbon dioxide concentrations and temperatures will continue to increase. Such changes will exert opposing pressures on the distribution of C3 and C4 plants. Higher CO2 concentrations will favor C3 plants, where higher temperatures will favor C4 plants. Nonetheless, the warming of Arctic regions, which is anticipated to be more extreme than in other parts of the world, will push wheat and corn belts further northward. Global changes in the availability of water and nutrients will also influence the distribution of C3 and C4 plants. The following video segments examine the interactions of these resources and plants. In summary, rising carbon dioxide levels are associated with decreasing oxygen levels. 2. The atmospheric composition anticipated by the end of the century will profoundly influence plant life, but will not cause respiratory distress in humans. Number 3. Carbon dioxide enrichment in the short term stimulates C3 carbon fixation, the biochemical pathway by which all photosynthetic organisms generate stable energetic compounds such as sugars. Number 4. Some plant species have developed a CO2 pumping mechanism known as C4 carbon fixation. Number 5. Interactions among C3 and C4 carbon fixation and climate change will determine where food is grown.